Hello everyone, in this class we'll try to learn about what are nerve potentials. Genesis of nerve potentials. The ability of cell to generate action potential in their membrane is known as excitability. Nerve is highly excitable tissue which can be stimulated by electrical, chemical and mechanical forms of energy. In the neuron, processing of information takes place chiefly in the cell body. The message transmission occurs by means of generation of propagation of electrical signals in the axons from one end to the other. There are different types of nerve potential. It can be graded nerve potential or action potential. Graded potential spread the signal over short distances. Action potential transmit the message throughout the length of the plasma membrane. Another type of response is seen in the neuronal membrane is called local response. So these are the different potential which we can see in the nerve. Okay, so they can be cat electronic potential or an electronic potential, which are negative potential. So cat electronic potential can sometimes lead to local response, but if it reaches the firing level, so then they will lead to a propagated action potential. A graded or electrotonic potential. Local, non-propagated potentials of small magnitude in response to depolarizing or hyperpolarizing stimulus of lesser strength. They are recorded from the membranes of dendrites and cell body. So they can be cat electrotonic or anelectrotonic. When membrane is electrically stimulated, the cathodal end of the stimulator evokes the depolarizing response is called as cat electrotonic potential. Anodal end of the stimulator evokes a hyperpolarizing response which is called as an electrotonic potential. Following the depolarizing graded potential, the repolarizing forces try to neutralize the disturbance in the RMP created by sodium entry. The potassium tends to come out of the cell and chloride enters through the leaky channels to maintain electrical neutrality. The sodium moves away by diffusion from the site of stimulus that decreases the amplitude of graded potential with the time. The increased activity of sodium potassium ATPase that pumps 3 sodium out, and all these lead to the gradual return of membrane potential towards the resting value. Properties of graded potential. The magnitude of potential change is proportionate to the stimulus strength. Then there can be decremental conduction. Graded potential decay progressively with the time and distance. The potential die out within distance of 3 millimeters from the site of stimulus. If recorded immediately after the application of the stimulus, the amplitude is larger, and with the delay recording, the amplitude is smaller. It can be depolarizing or hyperpolarizing nature. When the change in the potential is plotted against time, the graph appears like a wave. With application of increasing strength of cathodal stimuli, gradually rising upward waveforms appear. With application of increasing strength of anodal stimuli, the graph displays downward waveforms. If the second stimulus is applied before the potential produced by the first stimulus has disappeared, both the potential changes are added together, producing a larger or prolonged wave in the recording. So this happens due to the arrival of more sodium at the site of stimulus before neutralization of all sodium influx caused by first stimulus. Similarly, an electrotonic potential exhibit a property of summation. There are different forms of graded potential. They are named according to the membrane from where they are recorded. So end plate potential is recorded from the end plate area of skeletal muscle membrane. Receptor potential recorded from sensory nerve endings. Synaptic potential recorded from the membrane of postsynaptic neurons, and pacemaker potential recorded from the membrane of pacemaker cell in the heart, intestine, and so on. Local response. If a axon is stimulated with slowly increasing strength of stimuli, when the membrane potential is decreased by 7 millivolts, the pattern of graded potential is altered. The response becomes greater than what is expected for the strength of stimulus. So this enhanced response is known as local response. Similar to the graded potential, local response gradually dies out with increasing distance. Local response is seen with a depolarizing stimulus or lower strength and not with hyperpolarizing one. Ionic basis of the local response. As the applied cathodal stimulus is progressively raised from zero, the influx of sodium through the leaky sodium channel increases proportionately. Consequently, the membrane potential gradually decreases from minus 70 millivolts. As the membrane potential reaches minus 63 millivolts, few of the voltage gated sodium channels starts opening, which allows the entry of some more sodium. Thus, sodium influx through the leaky channels added to the sodium influx through voltage gated channels results a heightened response called as local response. So this is observed within the potential range from minus 63 millivolts to minus 55 millivolts. There can be different types of stimuli. At minus 55 millivolts, the neuron starts generating action potential. So this is termed as a firing or discharge of neuron, and minus 55 millivolts is known as firing level or threshold level. Threshold stimulus is the lowest strength of stimulus that elicits an action potential. Subthreshold stimuli less than the strength than the threshold. Suprathreshold is a stimuli higher the strength than the threshold. And subliminal stimulus is a sensory stimulus below a person threshold or conscious perception. Strength and duration. There are two important aspects of the stimuli. They have a complementary role in determining the excitability of the tissue, and they are inversely related to each other. Okay. So if strength is increased, duration for the excitability is reduced. If the strength is reduced, duration for the excitability will be increased. Okay. So this is the strength duration curve. Okay. So as you can see, so in this excitability curve, okay, so at a rho-based strength, the muscle is or the nerve is excited, and the time required for excitation is called as utilization time. Okay. So as and when you increase the strength of the stimuli, okay, so there will be gradual decrease in the time required for the excitation. Okay. And when the strength is increased to double of the Rho base, and the time required for excitation of the nerve is called as a chronaxy. Rho base utilization time and chronaxy. Rho base is the lowest strength of current that can elicit an action potential. Utilization time is the duration for which it must be applied to elicit action potential. A stronger stimulus requires less time to excite the tissue, and stimulus stronger than the upper limit may damage the tissue. Duration is long with weak stimuli and short with the strong stimuli. Chronaxy is the time required for a stimulus of double the Rho base strength to produce an action potential. So it is nerve has a shorter chronaxy, and it is more excitable than the muscle. Accommodation. If a slow depolarizing stimulus uh, is applied, the tissue fails to produce an action potential because it adapts to the stimulus. So this phenomenon is known as accommodation. Some of the voltage gated sodium channels starts opening at the membrane potentials of minus 63 millivolts, but they soon get inactivated before another set of channels open, because the membrane takes longer time to arrive at the next level of potential. Besides, slow voltage gated potassium channels open, allowing compensatory efflux of potassium, which opposes the depolarization to the critical firing level. So that leads to accommodation. Coming to the action potential, so action potential is a transient change in the membrane potential about 100 millivolts. It is characterized by gradual depolarization to threshold and rapid ascent to the membrane potential followed by the phase of repolarization. So, which is conducted along the axon, and it obeys the all or none law. Okay, and it has the same shape and amplitude. So this is also known as impulse or spike potential. So duration of action potential is in nerve muscle in a nerve is 1 milliseconds and amplitude is about 100 millivolts. So the action potential, they never summit. A definite interval exists before the second action potential is fired, and which is called as a refractory period. The latent period is interval between application of the stimulus and the onset of action potential. Action potential is always preceded by a latent period, and the duration of the latent period depends on the distance between the stimulating and recording electrode and the type and diameter of the nerve fiber. There are different phases of action potential. There are importantly three phases: the phase of depolarization, the phase of repolarization, and phase of after polarization, after hyperpolarization. Phase of depolarization consists of slow depolarization to the threshold, so which leads to local response, and then rapid rising phase, overshoot, and a peak. So then phase of repolarization includes rapid falling phase and slower terminal part, which is called as after depolarization. Then phase of after hyperpolarization includes brief period of undershoot and the return of the membrane potential to resting level. Okay, so these are the different phases of action potential. So where initially you will see the latent period, so then the local response, so which when it reaches to the firing level, it leads to the rapid rising phase, which is called as depolarization. So then rapid falling phase. Okay, so then after depolarization, okay, so then after hyperpolarization phase. Phase of depolarization. The depolarization phase is initially slow, begins with the takeoff from the baseline, that is resting membrane potential, to the firing level, and this is also called as local response. As soon as the threshold is reached, the membrane potential rises in a rapid and sharp manner in which the membrane potential decreases very fast, and this is known as a rapid rising phase. The potential then crosses the zero or isopotential level, then continues to rise rapidly to peak. Okay, so this is called as overshoot. The phase of depolarization goes up to plus 35 millivolts, and this point is called as peak. Then you have a phase of repolarization. The repolarization process starts as soon as the depolarization is over and is recorded as a downstroke. In the major part of the downstroke, action potential falls rapidly. This is called as rapid filling phase of repolarization. So this is followed by slower repolarization, which is the terminal part of the repolarization phase that brings the membrane potential to the baseline. So this is called as after repolarization. Then you have a after hyperpolarization. The phase of repolarization is followed by an after hyperpolarization phase. The membrane potential undershoots, it goes below the baseline, and then the membrane potential returns back to the resting membrane level. So what are the ionic bases for the action potential? The sequential changes in the membrane permeability is mainly by the sodium and potassium, so leading to large fluxes of these ions across the cell membrane and along with their gradients. So depolarization is mainly because of the influx of sodium, and repolarization is due to mainly the efflux of potassium. The voltage gated sodium and potassium channel contributes to the different phases of action potential. Depolarization, in the exon resting state, only leaky channels they open up. In the early part of the local response, due to the entry of sodium ions through the leaky channels, and when the change in the membrane potential is about just plus 30 millivolts, few of the voltage gated sodium channels begin to open at this point. At minus 55 millivolts threshold level, there is a simultaneous opening of the large number of these voltage gated sodium channels. They increase the membrane permeability to sodium by several hundred folds. So depolarization is due to the opening of voltage gated sodium channels. The in the stimulated region of the membrane at threshold, the number of sodium channels that have already opened cause simultaneous activation of huge numbers of sodium channels, which is called as autoactivation, which occurs very rapidly, and this leads to massive influx of sodium ions, producing a swift, large, and steep depolarization, changing the membrane potential to plus 35 millivolts. The opening of the few sodium channels leading to further opening of the sodium channels is called as a Hodgkin cycles, and this is an example for positive feedback control in which simultaneous triggering of an event further facilitates the process. Repolarization. This occurs due to the opening of voltage gated potassium channels marked by closure of voltage gated sodium channels, and thus they decrease the membrane permeability to sodium. So at the same time, voltage gated potassium channels are maximally activated, that increases the membrane permeability to potassium several times. The voltage gated potassium channels are sensitive to the same depolarization that opens the voltage gated sodium channels, but they open more slowly. Factors that favoring potassium efflux are potassium concentration is much higher inside the cell, which creates the chemical gradient. At the peak of the action potential outside the membrane is negative in comparison to inside, which is plus 35 millivolts, that is the electrical gradient. The rapid falling phase of repolarization is brought by decline in sodium influx together with increase in potassium influx. Once the membrane potential drops close to the isopotential level and moves towards the resting membrane potential, inside the membrane becomes negative, that it limits the efflux of potassium. So after depolarization phase is due to the slower exit of potassium ions, so that considerably decreases the rate of repolarization and makes the repolarization curve oblique or less steep. Following after depolarization, the membrane potential reaches the resting level. So at this level, though the most of the voltage gated potassium channels are closed, some of them still remain open and allowing continued efflux of potassium ions. As a result, membrane potential becomes more negative than the resting membrane potential, giving rise to the prolonged and slow undershoot, which is called as phase of after hyperpolarization. Finally, the potassium channels completely close, restoring the membrane potential to the resting membrane potential. So these are the opening and closure of the gates during the different phases of action potential. In the resting phase, both the potassium and the sodium channels, they are closed. During depolarization, sodium channels open up. At the peak of the action potential, the sodium channel closes and potassium channel begins to open, leading to potassium efflux. Okay. So in the later part of the repolarization, the potassium efflux slow down, and the later part of uh, the repolarization, then ultimately there will be closure of the potassium, which is going to lead to after hyperpolarization. Okay. So again, this is at the resting state where the sodium channel is open. Okay, and at the peak of depolarization, the sodium channels, they're going to open, and uh, and at the peak of the depolarization, inactivated gate of sodium channel, they're going to close and they stop potassium sodium influx. So this is the ionic conduction during the action potential. Okay, if this is the action potential, okay, so this is the rate of the activation of voltage gated sodium channels. Okay, so they come to the peak and then gradually decrease. Okay, and this is the opening of the potassium channels. Okay, so which occurs immediately after uh, uh, the peak of the action potential. So it gradually increases. So then at the during the late phase of repolarization, the potassium efflux is reduced. The rate of potassium efflux is reduced. At the end of the action potential, more sodium and less potassium inside the cell. The ionic composition is restored by the increased activity of sodium potassium ATPase. About 1 of every 1 million sodium ions go into the cell, and about the same number of potassium ions come out of the cell during action potential. So this produces negligible change in the intracellular ionic concentration. However, if this imbalance were not taken care of following repeated generation of action potential, slowly the concentration gradient of sodium and potassium across the membrane will cease to exist. Effect of extracellular ionic changes. When extracellular sodium concentration is decreased, the amplitude of action potential becomes smaller than the usual because of the concentration gradient for sodium that drives the sodium into the cell is reduced. When the extracellular sodium concentration is increased, the amplitude of the action potential may increase. When extracellular potassium concentration is decreased, the membrane potential become more negative as the resting potassium efflux is favored by the increased concentration of gradient across the cell membrane. And when extracellular potassium is increased, the membrane potential come closer to the firing level and the membrane becomes more excitable. When extracellular calcium concentration is decreased, the electrical potential difference across the cell membrane is decreased as the inside becomes less negative compared to the outside. The resting membrane potential come closer to the firing level so that the magnitude of depolarization needed to reach the firing level is less. Hence, the decrease in extracellular calcium concentration increases the excitability of the tissue as observed in hypocalcemic tetany that occurs during hypoparathyroidism. If the extracellular calcium concentration is increased, the RMP goes away from the resting from the firing level, decreasing the tissue excitability. So these are some of the ion channel blockers. Okay, so the, the blockers which blocks the sodium channels are local anesthetics like lidocaine, procaine, toxins like tetrodotoxin found in ovaries of pufferfish and in the tissues of salamanders, and saxitoxin is accumulated in the tissues of the shellfish. Some of the potassium channel blockers are tetraethylammonium, that is TEA, and 4-aminopyrin. The, the chemical which blocks the sodium potassium pump is dinitrophenol. In an action potential is formed, it appears with the maximum size and shape, otherwise it does not form at all, and this is called as all or none law. A threshold stimulus fails to excite the tissue, and only a stimulus of threshold magnitude elicits an action potential. If suprathreshold stimuli are applied, the action potential resulting from them have the same amplitude, duration, and form as produced by the threshold stimuli, provided the experimental conditions remains the same. The mechanism, the activation of the gates of voltage gated sodium channel open as soon as uh, plus 15 millivolts of depolarization is achieved. Following that, extra degree of depolarization is of no further use as the membrane automatically achieves the another 100 millivolts of depolarization. After the threshold level reached, the amount of sodium influx becomes independent of the stimulus factor. The number of voltage-gated sodium channels over the external membrane of unmyelinated axons as well as the nodes of Ranier of myelinated axons remains fairly constant. Once the action potential is formed, it appears with the maximum size and shape, otherwise it does not form at all. The action potential occurs with a constant amplitude and shape whether the stimulus is of threshold or supra-threshold magnitude. Okay, so this is the diagram showing uh, the the property of all and none. Okay, so when subthreshold stimuli is given, there will be no production of action potential. So when threshold stimuli is given, there will be production of action potential, and the suprathreshold stimuli, the action potential amplitude is going to remain the same. During the action potential, the stimulated area of the membrane happens to be unresponsive to second stimulus in the most part, and later it requires a greater degree of depolarization to get excited again. So refractory period is the length of the time during which the membrane is unresponsive to the second stimulus. So there can be uh, absolute refractory period or there can be relative refractory period. Absolute refractory period is the period of action potential during which application of second stimulus of any strength and duration does not produce another action potential. And from the time of the firing, so this corresponds to the time of the firing level is reached until the repolarization is about 1/3 complete. Okay. So this is the phase of absolute uh, uh, refractory period, and this phase is relative refractory period. Okay, so this time is between the firing level and the 1/3 of the repolarization phase, and after 1/3 of the repolarization phase, it is a relative refractory period. The mechanism of absolute refractory period. At the peak of action potential, the inactivation gates of the voltage-gated sodium channels close and they remain in that inactivated state for the some time before returning to the resting state. So these sodium channels can reopen in response to a second stimulus only after attaining resting state. So importance of absolute refractory period. This limits the number of action potential that the nerve can fire in a given period of time. So ARP determines the rate of discharge of a nerve fiber. The usual firing rate is 10 to 1000 impulses per second, and in large diameter nerve fibers, the ARP is zero point 0.4 milliseconds. Firing rate is 2500 impulses per second, and in small diameter fibers, the absolute refractory period is 4 milliseconds. Firing rates of 250 impulses per second. And ARP is also responsible for one-way conduction of action potential. Relative refractory period is the period following ARP during which application of suprathreshold stimulus can elicit second action potential. This starts from the end of the ARP to the start of after depolarization. All the sodium channels present at the site of stimulus does not achieve an inactivated state or resting state exactly at the same time. By the time of relative refractory period, some of the channels have returned to their initial resting stage and they can open in response to second stimulus. Suprathreshold stimulus can spread to a larger area over the membrane and open more number of voltage-gated sodium channels. Okay, so that leads to the formation of one more action potential. So the initiation of action potential, the first node of Ranier in sensory neurons, and it occurs in the initial segment of the axon hillock in the motor neurons, and these areas are known as trigger zones. They have very high concentration of voltage gated sodium channels and potassium channels. The synaptic potential generated at the dendrites or the cell body is integrated by the cell body and transmitted to the axon hillock. In this potential, if, if this potential is sufficient to depolarize the membrane of the axon hillock to firing level, the membrane is the membrane easily fires an action potential. So once formed, the action potential is regenerated at the regular intervals to be transmitted from the initial segment to the axon or to the axon terminal. The speed of conduction of the impulse depend on the myelination diameter of the axon, and myelination, the conduction velocity is more in myelinated axon and is proportionate to the degree of myelination. So it also depends on diameter. The conduction velocity is proportionate to the diameter of the fiber. Large diameter fiber, faster rate of conduction occurs, and large diameter fibers have less cytoplasmic resistance. So the flow of ions across the membrane is easier. Propagation of action potential in unmyelinated axon. At the site of genesis of action potential, large influx of potassium charges, potassium positive changes into the membrane occurs, which is called as current sink. The positive charges diffuse away from the site of accumulation. The adjacent membrane, which is in its resting stage, has a potential of minus 70 millivolts. This potential difference allows the positive charges to flow towards the adjacent negative area. Consequently, the potential of the adjacent membrane decreases and reaches the threshold value as the fraction of sodium ions that move to the nearby negative area are sufficient enough to bring the adjacent membrane to the firing level. Okay. So this is how propagation of the nerve impulses occurs, so which is called as current sink. Okay. So when the area is depolarized, okay, so there will be presence of lot of positive ions. So these are going to move towards the negative ions, and they are going to produce another the firing potential here, and then the action potential in the adjacent area, and that is how an action potential is propagated. So this results in the opening of the voltage gated sodium channels in the present in that area, firing an action potential. Similarly, from the site of second action potential, positive changes flow to the adjacent resting membrane and decrease the potential to the threshold level. So this activates the voltage gated sodium channels present in that membrane, resulting another action potential. In this manner, each point of membrane gets depolarized to the firing level and produces action potential. As the depolarization and repolarization phases of ensuring action potentials go on, there is a sequential opening and closing of the potassium, sodium and potassium channels along the axonal membrane. So in myelinated portion of the axon, there are few voltage gated sodium channels. Myelin sheath acts as an insulator and does not allow free flow of ions across the membrane. The concentration of the positive charges does not decrease fast because of the less leakage. So this helps the changes to spread further along the axon. So this is what is going to happen in the myelinated fibers. The action potential, they are going to jump from one side to another side, and which is called as the saltatory conduction. The local current travels like a graded potential and dies away 37% of its maximum strength over the distance of about 3 millimeters. At the internodal distance is 1 to 2 millimeters, the local current definitely arrives at the adjacent node of Ranvier and decreases its membrane potential to give another action potential. The voltage gated sodium channels are present in the large numbers at the node of Ranvier as soon as the nodal membrane gets depolarized to threshold level, the action potential quickly fired. The action potential is generated at each node of Ranivere. Okay, so this is called as saltatory conduction. So in the Latin word, salta means to jump. The action potential rapidly proceeds from one node to the next and pause at each node to get generated. Advantages in myelinated axon. In myelinated axon, velocity of conduction is faster. So it also helps in conservation of energy. Since the ionic flux occurs only at the nodes of Ranivere, the total membrane area across which ionic balance has to be restored is much less in uh, unmyelinated axon following propagation of action potential. Activation of larger number of sodium potassium ATPase and higher expenditure of the energy. In motor neuron from axon hillock towards the axon terminal, in the sensory neuron it is from the first node of Ranivere towards the central nervous system. Action potential does not travel from axon back towards the trigger zone following depolarization. The area on the membrane where action potential was produced becomes refractory. So, so it can spread in both the direction along the axon if it is initiated between the trigger zone and the axon terminal. So these are some of uh, the properties of the nerve fibers.